you very much. It's a pleasure to be here. Right, so I'm going to talk today about protein structure prediction in a sort of a post-AlphaFold2 world. And I know, of course, this is very topical at the moment. So what I'll do is I'll spend some fraction of the time talking about AlphaFold2, about what it sort of can and can't do, and so just to contextualize where we are. And then I'll step back actually for a bit and then sort of place AlphaFold2 within the broader context of the last, say, 10 years or so in terms of the various method development that's happened in this space and really sort of what led to AlphaFold2 in a way. And that would maybe take about half the talk. And then the remaining half, I'll tell you more about some of the work that we've been doing in the space, trying to kind of go beyond AlphaFold2, addressing some of its limitations, and hopefully, sort of wrap up there, and then take any questions. And I'm happy to, I know AlphaFold2 is on a lot of people's minds, so I'm more than happy to, answer any questions about it if people are interested, beyond our own work as well. Okay, so there very much has been this revolution, this deep learning evolution, and it's really impacted, virtually every field in computer science, certainly in machine learning, and protein structure prediction is one of them. So if you're not familiar with the field, there's this competition called CASP, happens every 2 years, has been going on for over 20 years, where people predict structures in a blind fashion, structures that have been determined but they've not yet been publicly released. And there are a couple of categories in CASP. One of them is the free modeling category. This one, which is the kind of the hard targets. These are the ones that don't look like anything in the PDB. And if you look at this space, essentially, I don't have it fully plotted here, but basically from like in the mid 2000s till about 2016, really there was not a ton of progress. It was just fairly halting. There were a few, there were important breakthroughs that have happened on the 2010s having to do with this idea of coevolution, but they didn't sort of quite percolate to the top of CASP up until 2016. And then the first AlphaFold came on the scene. That was in 2018 in CASP 13, and that really I think made a very significant sort of leap in terms of prediction accuracy. But in some ways, that leap, one could argue, was very much from a methodological standpoint, was somewhat incremental. Did not sort of introduce any fundamentally new concepts. It was really more about sort of engineering. It was about taking the ideas that were out there and then sort of improving them in a way and tuning them in a way that made AlphaFold significantly better than sort of the competitors. And I think it's fair to say that for many of us at the time, we thought that sort of going beyond this would be quite challenging. That the low-hanging fruit has been picked, and that would require some degree of innovation to really be able to push that beyond the kind of the 6 angstrom, roughly line, which is where these methods were at the time, including AlphaFold. So I think that the surprise and really, what I think kind of shocked everybody in the space is the fact that DeepMind sort of managed to execute on this and really sort of do deliver the innovations that were required to go beyond, that sort of 6 angstrom line that was in 2018 to something that I think in some real sense begins to rival experimental methods. It certainly doesn't obsolete them in any way or shape, but it certainly begins to, at least for certain classes of questions, for certain kinds of problems, begins to offer a compelling alternative to having to go to get a crystal structure. And this is something that I think is very exciting, obviously, and I suspect will support and kind of motivate much new developments in the field in the years to come. So just it's good to kind of step back for a moment and just think about what AlphaFold2 can and cannot do, because it's not sort of omnipotent. it does have some limitations. It's I would say, at least in the context of CASP 14, which is really the only sort of, I would say, kind of rigorous assessment that can be done. It was primarily tested on single domain apoproteins. In principle, up to several thousand residues, but obviously, given that they're single domains, they're not usually that big. And here it achieved a median accuracy of about 2.1 angstroms. And here you can see the distribution of all its error. So half the time was about 2.1 angstroms. Up to about 90% of the time, it was well within 4 angstroms, which is pretty impressive. With a few instances where it did poorly, but at least some fraction of these can be explained away by either having sort of intrinsically disordered regions or having folding that really depends on other proteins or other ligands being present. So the number of sort of genuine failures were quite limited in CASP 14, and obviously now we've seen it been applied to essentially every protein in human, and it cannot predict for maybe about 50% of proteins well-ordered structures, but that's because this order is a very abundant phenomenon in proteins, and I think AlphaFold2 sort of communicated that. It sort of made it very visible, the fact that there's so much disorder in proteins. When you're used to sort of studying at PDB structures, you only see the ordered regions. And this gives you a sense of how well it does, which is to say very well. It does have some sort of interesting kind of characteristics, or maybe one even can call pathologies. This is one example of that showed up in Cas14, and this has been the case in what people have tried to use it for oligomeric structures, which is that if there are strong interdomain contacts, say within a sort of a trimer, for example, as we see on the left here, AlphaFold actually recovers that trimeric structure and does so pretty well. But the funny thing is, at least in the context of CASP, the task that was presented with was predicting monomeric structure. So it's sort of able to recover the trimer, but it's in a way sort of shouldn't do so, right? Because the monomer, if one is really interested in the prediction of the monomeric structure, it should give you something completely different. So this is an instance where it sort of does the right thing for the wrong reasons in a way, and I think we're seeing that in multiple cases. I'll show you another one here, where in this particular target, it sort of gets the side chains really very accurately, the active sites, but that active site requires a zinc ion for it to be stabilized, and obviously AlphaFold2 is sort of not aware of zinc ions. It's quite all it knows are just the natural amino acids. Yet it was sort of able to place things correctly, and so again, if the task was to predict the structure without the zinc ion, it should have presumably predicted something different. But because it sort of can almost impute that there ought to be some sort of ion there, it's sort of able to give you what it expects to see in the PDB. And so in some ways, it's sort of a PDB structure predictor as opposed to kind of a structure predictor. Now, it does, I think till now, struggle a bit with multi-domain proteins. This is one example of that. But I think one thing that's becoming increasingly clear is that many of these multi-domain proteins do have a lot of inherent flexibility. And so when we see a particular snapshot that AlphaFold2 kind of generates, it's I think hard to say at this point whether that structure, that conformation, really reflects an error, assuming that conformation sort of disagrees with what one sees in the PDB. It's a bit hard to say at this point whether that's because it's erroneous or because these proteins are flexible. And I think this is kind of one of the interesting things as we begin to wrestle with so the idea that these predictions are accurate is how do we reconcile this agreement with experiment, and how do we move forward, maybe even more importantly, in terms of characterizing these proteins in a way that really goes beyond single structures. One area where it's not been tested with are things like mutations or design proteins. Although people have been doing things like this sort of unofficially, but there's not been kind of a rigorous assessment, and it really can't yet handle single sequences, so without a multiple sequence alignment, or things like complexes, again at least officially. Although people have been using it in also sort of unofficial ways, and things like ligands, cofactors, etc. So that's sort of where we currently stand, and I think it's fair to say, like I alluded, I think it's incredibly impressive. But there do remain some blind spots that I think we would like to sort of be able to tackle, and I'll talk about those in a moment. But before I do that, what I thought would be good to do is just step back for a moment and kind of consider an algorithm space for protein structure prediction, and sort of place AlphaFold2 and other schemes in this algorithm space before we begin to think about where do we need to go next, and what are the kind of the outstanding problems that remain. So there are many ways that one can sort of imagine laying out this algorithm space. The way I do it is to think of these two primary axes. One is sequence information, so how much data essentially is going into a prediction task. So this is really information at inference time. This is not for training, but this is for inference, for actually making a new prediction. And then the y-axis is accuracy, is how well do we do. So I think it's fair to say that for the longest time, people thought that the way to get at this problem is really using physics-based approaches, trying to kind of emulate the physical process of folding, or at least energy sampling. And so things like systems like Rosetta or ITAZA, which existed for a long time and still do, really sort of try to tackle this problem by essentially taking a single amino acid sequence and trying to predict its structure. And I think it's fair to say that sort of purely physics-based approaches do struggle with this. They don't really work very well, and so they're on the lower end of the spectrum. Of course, things like Rosetta and ITAZA have had many elaborations now that involve machine learning and particularly coevolutionary information, and those work much better. But here I'm just focusing on the kind of purely physics-based components of these methods. Now, there was work on sort of adding templates, so it's essentially looking at the PDB for homologous proteins and using these as starting points. And when one does that, the predictions do improve significantly, even though even these physics-based predictions, in part because let's say, the global search is very difficult to do. But if you have a template that places you roughly where you need to be in structure space, then these methods can then sort of take you down to the kind of the refined structure. And I'll actually sort of illustrate a point about this in our own work in a moment. But suffice to that, without templates, at least things don't work very well. So to move, kind of fast forwarding a little bit to the early 2010s, this idea of coevolution, which has been around for really decades, finally began to work. And I'll talk about this in a moment. But the basic notion here is that you search the kind of sequence space, the sequence databases, and you identify proteins that are homologous to the one of interest. And you build a multiple sequence alignment out of those homologous sequences, and then you essentially try to identify residues that appear to be co-evolving. Either as one changes, the other one seems to change in concert, with the idea that this kind of constricted coevolution is indicative of perhaps spatial proximity in 3D space. So some of the early methods really didn't use any machine learning, or didn't even use any kind of physics-based refinement. They simply just took this idea and see if they could recover the structure using almost like NMR relaxation kind of techniques. And so they sort of worked, but sort of didn't. They worked when you have very deep MSAs, but otherwise they had somewhat limited applicability. An important sort of shift, I think, happened in 2016 with the first RaptorX that was developed by Jim Bouzoux's group, which took this idea of kind of coevolution, but then kind of reformulated it as a supervised learning problem. So all these approaches, prior to RaptorX, were about sort of unsupervised learning, simply just trying to recover the covariance structure and from that for the protein structure. But RaptorX sort of kind of turned the problem in a different way, saying, okay, well, we have structures in PDB, we have these sort of covariation matrices, can we put the two together and then formulate this as a supervised learning problem to predict structure from these multiple sequence alignments? And that really kind of led to, I think, a sizable improvement in accuracy. And so this is, I said DL. So there's so here now it's deep learning, it's supervised learning, it's something which is learning from actual structural data. This was followed by RaptorX 18, which sort of went beyond simply predicting contact maps, which is what these other approaches were doing, but actually predicting distances between residues, and then using those information essentially as constraints to physics-based approaches to follow a protein. This was sort of, I would say, contemporaneous with AlphaFold1. In fact, I think the preprint for RaptorX 18 came out just before AlphaFold, although, they were obviously developed independently. AlphaFold certainly worked better than RaptorX 18, but I would say the kind of the conceptual underpinning was very similar. It was the same kind of piece of innovation. They were both working off of predicting distances, but AlphaFold was a bit more refined in the way it did so. And this sort of, kind of triggered follow-up work. Rosetta was one of them. Which was sort of, which was in this general vein of combining coevolution ideas with sort of physics-based refinement and post-processing. Sort of just before this work, I had developed a method called RGN, which did a couple of things. So first, it's did not use coevolution, but it didn't just use single sequences. It sort of went somewhere in between. It used position-specific scoring matrices. So basically, you could think of these, if coevolution is about second-order information about pairwise interactions between residues, PSMs are just sort of first-order information. There's just about the propensity of any given residue to mutate to any other residue, essentially. So it's sort of a simpler construction. So it has less data in a way in it. And my motivation here was to move toward the single sequence direction, but maybe not take the leap all at once. So that's one aspect. But I think maybe the more important aspect here was that this was sort of the first method to really be end-to-end differentiable, right? So this was, all these other approaches, that I'm showing in the third column, the coevolution approaches, they did not predict the structure directly. They predicted some sort of proxy object, like the distances between residues or the contact map, whether two residues are in proximity or not, and then fed that into, like I said, a physics-based engine to follow the protein. The RGN method, on the other hand, was end-to-end differentiable. It went from the sequence or the PSM to the structure, all using kind of differentiable neuralized components. And this, I think, was an important shift into how we build these systems, because it allows us, as I'll talk about in a moment, to kind of optimize the behavior of the system from end to end. There was another method, again, developed contemporaneously, came out of there on the same time, called NEMO, similar idea. And in fact, NEMO tried to push this toward the single sequence direction, suffered a performance penalty as a result, but still sort of was really trying to push this idea of going from a single sequence to the structure. Now, fast forward to last year, RaptorX 20 came out, which was sort of an elaboration of TR-Rosetta, again, worked a little bit better, and actually tried to go to the PSM route. So tried to kind of, again, move in the leftward direction, did better than RGN and NEMO, but was not actually end-to-end differentiable, but it just simply, used the kind of the existing paradigm, but worked off of PSSMs. Then, of course, AlphaFold2 comes out, and AlphaFold2 sort of does really many things. But one of the things it does is that it uses a full multiple sequence alignment as opposed to kind of try to extract covariation information. So it does so implicitly, presumably. The network learns to extract covariation, but as a fundamental object, the thing that AlphaFold2 operates on is really the full multiple sequence alignment. And this in part explains its performance gain, but not really very much. I mean, much of surprisingly, when DeepMind's gone and ablated different features of AlphaFold2, when you remove their full MSA, that doesn't seem to impact performance all that much. So there were other architectural innovations that definitely contribute to its performance. One of which is the fact that it is end-to-end differentiable. So AlphaFold2 was sort of the first method that combines kind of the idea of covariation or say multiple sequence alignments with end-to-end differentiability, and that I think also played some significant role in its performance. But stepping back now for a moment, where I think the holy grail is, and both because I think there are certain applications which rely on this, but also because I think if we want to understand protein folding in a detailed fashion, we would like to be able to go from a single sequence to the structure. And so to me, that's really the holy grail, being able to be as good as AlphaFold2 while predicting from single sequences. And so this is a direction that we've been sort of pushing, and we certainly haven't achieved it. The performance is more along the lines of the first AlphaFold than the second AlphaFold, but we are able to do some single sequences, which I think is quite exciting. And so I'll tell you more about this now. So coming back to kind of the algorithm space question. So just to decompose it a little bit, there's really, I would say, kind of four main components in any of these systems. One is the input, what goes into the prediction. There's the torso, the thing that kind of does some sort of reasoning. There's the output that generates that represents what is generated from that torso, and this output may or may not be the final 3D structure. And if it's not the final 3D structure, then there's a post-processing component which takes that output and then actually uses some sort of physics-based engine or something else to generate the 3D structure. Now, from our perspective, what really matters here is that what gets optimized within kind of a machine learning framework is that output, right? So if your loss function, the things in which the optimization procedure is defined, that's all based on the output, right? Not based on the pink stuff. And so, and this is important for several reasons. But when one speaks of an end-to-end differentiable approach, it's really about what is the output and whether the output includes the 3D structure itself. Because that is the part that actually gets differentiated through in a kind of an optimization framework. So coming to kind of these methods, the ones that I talked about earlier, the kind of covariation approaches, the way these work basically is that, again, you take a multiple sequence alignment, you generate a structure, maybe say a contact map. No, I take it back for a moment. For the input side, you don't generate a contact map. You generate something that looks like a POTS model, something that encodes the covariation information, that then converts into something like a contact map on the output side. And then things like AlphaFold or TR-Rosetta refine that to make it more about distances as opposed to just binary contact. In fact, TR-Rosetta not just includes distances, but also include angular information between residues. So sort of richer outputs, but still proxies to the structure, to the things that one wants. And in particular, all of these approaches, including the first AlphaFold, relied on post-processing to take that kind of construct, that kind of artifact, and convert it into a final three-dimensional structure. And this can be very slow. Incidentally, this could take potentially tens of thousands of hours, these physics-based engines. But more importantly for our discussion, it sort of prevents optimization of the final structure, right? So if you're trying to build something which is sort of optimized, in terms of all of its parameters to behave in a certain way, you want that behavior to be tied to the final structure as opposed to some sort of proxy quantity. But that piece was missing when one had the post-processing step. And in particular, is one way in which this is particularly problematic, which is that the outputs can be self-inconsistent, meaning that you, for example, can have multiple distances which cannot simultaneously be embedded in 3D space. This is sort of the general problem. If the distances are just simply predicted by a model that does not enforce a triangular inequality. And so this lack of self-inconsistency is sort of an inherent issue with these non-end-to-end differentiable models. So now moving to kind of the end-to-end differentiable models, the ones that I developed and others, the idea here is to predict the structure directly, and then by construction, since you are predicting the structure, then it is self-consistent. So what I focused on at the time was really, like I said, sort of two pieces. One is to formulate the output in a way to be end-to-end differentiable. And the way we did this was to essentially kind of suppose that there's a limited alphabet of torsion angles. So not an amino acid alphabet, but a torsional angle alphabet in torsion space. And then what we do is, so we essentially predict probability distribution over that alphabet, and then project the letters of this alphabet into essentially kind of a, well, the technical details are a bit involved, but basically project them onto a circle essentially, and then take the average of the points on that circle that corresponds to the different letters in the alphabet, weighted by the probabilities that were predicted by the model. And this essentially generates sort of a new point and therefore a new angle that is a sort of a weighted mixture of the angles that are predicted by the model. So this is a bit of a roundabout construction, but the reason this is done is to sort of address some issues that have to do with the optimization of this model. I'm happy to go into this later on. But just suffice it to say that so we need this construction to make this model easily optimizable, to sort of allow the gradients to kind of flow smoothly through the model. So we do this, and so essentially at every residue, we're predicting three torsion angles. And those torsion angles essentially describe the geometry of around a single residue. It's actually so it's not just the torsion; it's also the bond rotation and the bond extension, the bond length. But those quantities are fixed, so we don't need to predict them. But all of those go together in this particular sort of formulation, the one that's shown on the right. And by doing so, then we're able to kind of recover the whole structure. So building it one bit at a time, one atom at a time. And so this literally kind of sequentially just builds up the whole protein. And does so differentiably, so we could backprop through this entire process. Now, I mentioned this earlier in the kind of the first construction of the RGN. It was not really a single sequence. We still use a multiple sequence alignment, but that was turned into a position-specific scoring matrix, which was then used as the input. So it was somewhere in between those kind of two extremes. And the torsos, there are many different options. At the time, we had used an LSTM or a current neural network. Now, much of the focus in this space is really on sort of attention-based architectures like a transformer. Just to kind of just quickly mention, though, so in this approach, this very first approach, the predictions, were okay, as you see here, but we had some pathology. So you see, for example, the helical region, the one in the middle, even though it's sort of globally well predicted, locally the helices weren't very good. They sort of had poor local structure. And this was sort of a characteristic aspect of this kind of this architecture that we sort of tried to overcome in kind of the new version that we just actually preprinted a couple of weeks ago. So let me tell you about this new version now. So first, we kind of reformulated the geometric piece to work differently, to use what we're calling kind of a discrete Fourier construction, which sort of just focuses on the C alpha atoms and essentially describes them using kind of relative reference frames that relate one to the other, essentially. And I won't go into the math, but the point here, what the nice thing about this is that it sort of results in this construction in which the atom extension procedure I was showing you earlier reduces to simply matrix multiplication. So it makes it much faster, for one thing. But the other thing is that this makes it more, it requires fewer parameters, and by construction enforces that the local geometry of the protein is correct. So that you don't end up with these sort of weird pathologies that I was showing you earlier, where you have overly wound alpha helices, for example. So that's one bit. But maybe the more important bit was that we now actually go from single sequences. And the way we do this is we use a language model that allows us to essentially learn kind of a latent representation of sequence space that is then sort of sufficiently rich to do predictions from, or to make predictions from. So I suspect most people here are familiar with this, but if you're not, these language models are incredible. So this is one example, it's called GPT-3, where you give it a prompt, and that's the prompt that's shown in dark gray, and then you just have it generate sort of flowing text based on that prompt. And the way this method works is really quite simple. I'll describe it in a moment. So the basic idea is you give it a sequence, but then you mask some residues of that sequence. You sort of hide their identity. And then you feed this to a series of in this case, transform-based attention layers, and then ask the model to recover the sequence that was masked. So just simply kind of fill in the missing pieces. And this is done for natural language. This is what I just showed you a second ago with GPT-3. But we and other groups have applied this in the context of modeling proteins. And what you end up with is something that is a latent representation for each residue. So each residue is sort of embedded in this kind of latent space. But this latent space has inherent semantics, right? So because it's sort of learned kind of fill in these missing pieces. It learns by definition or by construction, it learns to essentially infer context. You know, that variant that's missing, it's not just a variant anymore. It sort of knows about the residues around it, and that context is what allows us now to take each of those residues and essentially extract more information out of them, as opposed to if it were just a one-hot encoding where all we knew that was just a valine. So that's one, but we actually do other things. We, for example, splice into different proteins, and then we ask the model to tell whether the splicing is the same, or is it an actual protein, or is it a spliced protein? And we found that this sort of makes the model learn global properties better. So that's essentially it for the anti-artificial part. Now, we do in this context still, you know, so we can predict the structure directly, and that's fine, but it doesn't always work perfectly. And so we do sort of provide a post-processing step if one wants higher accuracy structures. And this definitely suffers from the same kind of speed limitations that these other approaches suffer from. So we pay that price, but at least sort of an optional option that one can rely on if one is seeking to get more accurate structures. So let me now show you some results from. First, I'll show you some results, kind of comparing the first approach to this you know, improved approach, and then I'll show you some results comparing you know, AlphaFold2 and some of the other methods we have to our approach. So the first target this is actually a Cas14 target. So this is what had happened in December 2020 when we had those predictions done in a blind fashion. Here I'm actually showing it of TR-zeta. Both are quite similar to AlphaFold2, so it doesn't really matter. So the TR-zeta is in blue, is in cyan, and then our prediction, the ground truth is in white or gray. And here I'm showing kind of the very first RGN model, you know, very poor prediction in this case. And with RGN2 we see, you know, a substantial improvement, you know, halving the RMSDs. So that's good, but still far away from TR-zeta. But this is all pre-refinement. Now, if we include refinement, physics-based refinement, we actually can get the structure now to so well within striking distance of the experimental structure. And the point here, obviously, you know, so sort of the fact that we're able to predict this correctly is not in itself notable, given that TR-zeta kind of predicted well. What is notable here is that we're working off a single sequence, so we're not using coevolution information, and that's really the kind of the key thing. This is the point that I mean, this is the kind of major innovation, I would say, of this approach, that we're able to do this with single sequences. So that was just kind of one example showing you before and after, but let me show you now some general targets from Cas14 versus AlphaFold2. So these were, I would say, are some are better examples. These are cherry picks to kind of give you a sense of how well this works. You know, when we are comparing it to AlphaFold2 in the best of circumstances. Here, I should say, AlphaFold2 is using multiple sequence alignments, and these proteins have deep multiple sequence alignments. So this is not AlphaFold2 in a difficult territory. This is sort of, you know, normal territory. But you see here that and you see here we're not doing it clearly as well, but we're in the sort of the general vicinity. But in general, we do have more, I would say, middling performance if you're looking across the board. So these are more representative examples where you see here, a much bigger delta. What's interesting, this region in particular is somewhat well predicted by both our methods, but they're both about 4.2 exons. But then the pod that forms a multimeric beta barrel, that's actually poorly predicted by both methods. So they both end up doing poorly. But this is not surprising because this has again to do with kind of complexification, which neither method is sort of directly trying to address here. Now, when we look at experimental structures, just again giving a sense of what we saw in Cas14. Again, I mean, this is, I think, is more representative. So sometimes, so maybe on average we do something like 5 or 6 angstroms. So far from where AlphaFold2 is, but again, based on single sequences, which I think is quite interesting. This is sort of a funny example in that initially we saw that the prediction looked very similar to TR-zeta, which excited us because we thought we sort of nailed it. But then the real structure came out, and neither of us, neither of us got it. We were both quite far away, but AlphaFold2 really did nail it. So this is at least one example, just for full disclosure, where AlphaFold2 really does seem to do, qualitatively better than any sort of the existing approaches, at least at the time of Cas14. Since Rosetta Fold has sort of, it's sort of, which is a derivative of AlphaFold2, has closed some of that gap with respect to TR-zeta, although not entirely so, but certainly closed partially that gap. And incidentally, this particular example I'm showing here, it's sort of interesting because you can see, so I should explain. So this is the percentage of residues predicted, and this is sort of how far you are from the correct position. So the ideal prediction would be something that is, you know, a straight line at the bottom, then just goes vertically up. And AlphaFold2 is this line, so, you know, it's doing quite well. All methods are essentially doing about equally bad, and that's the example I just showed you earlier, with the exception of one other approach which does seem to do somewhat better. I don't remember which group this was from. Maybe it was another company, maybe Baidu or Tencent. But this approach was based on templates. So actually templates helped here, which is sort of interesting. And AlphaFold2 does use templates, so that may have been partly responsible for its performance gain. Now, so what I showed you just now are sort of cherry-picked examples from CAS14, you know, giving you a sense of how well it works or doesn't work. But where we think this approach is interesting and is sort of legitimately, you know, an improvement with respect to AlphaFold2 are sort of two categories of proteins. One are orphan proteins, so proteins for which we have either no multiple sequence alignments or essentially, well, yeah, I mean, essentially no multiple sequence alignments. So it's the instances in which we have just a single protein sequence, and that's all we have to work with. And the other are things like de novo proteins, proteins that we want to design for various purposes, which again by construction would not have been seen elsewhere. So now I'm showing here the results on comparing or assessing AlphaFold2 and RGN2 on orphan proteins. So just to kind of orient you, so we're using two metrics, GDTS and DRMSD. And so when we're in the top left quadrant here in the sort of dark greenish region, this is the region where we do better, where RGN2 does better on both metrics. Conversely, in the kind of the bottom right, the dark blue, this is where AlphaFold2 does better on both metrics, and then sort of split in the middle. So in terms of this region, at least, we do about, you know, 58% of the time, RGN2 is in this, you know, 58% of the predictions RGN2 is better on, and 11% AlphaFold2 is better on. So this is one instance where we seem to be doing significantly better, you know, than AlphaFold2 because of the fact that the multiple sequence alignment is non-existent. And this is certainly an application, you know, in plants and many other in rapidly evolving viruses, things like that, where being able to predict orphan proteins is quite critical. De novo proteins is a bit more mixed, but it's sort of interesting. So here I'm splitting the results based whether it's alpha helical rich or beta strand rich. And with alpha helices, we actually do better, even on de novo design proteins. Again, kind of similar to the previous situation, maybe even a bit more lopsided. On beta rich, AlphaFold2 does do better, and this may reflect the fact that's sort of able to kind of better capture long-range interactions in a protein. The AlphaFold2 has the structure module, which is essentially, you know, so trying to find the structure and find and that operates directly in 3D space. And we think that's a good idea, and we suspect that has in part to do with its ability to sort of see long-range interactions, perhaps. So this is one area where because the way RGN2 is set up, which is in some ways much more local, because it's having to essentially kind of build up the protein structure sequentially. This is one area where it may be suffering as a result, and we see this delta. Now, one interesting thing here, though, which I haven't said so far, is that all those proteins I'm showing you here were not in the AlphaFold2 training set. Now, we did do an analysis on a subset of proteins which were in the AlphaFold2 training set, and here's quite different. AlphaFold2 dominates much more so in the beta-rich regime, and it is almost tied with RGN2 in terms of the helical-rich proteins. So this is interesting, and I think it suggestive, at least. So I don't think this is a particularly legitimate experiment, because these proteins are in the AlphaFold2 training set. But it does seem to suggest that there is some overfitting going on with AlphaFold2, that we see this big discrepancy between this set, which is not in the training set, and here, which is in its training set. And this is, I think, important because I know many people have sort of looked at AlphaFold2 predictions and compared them to PDB structures that were in the training set. And sort of we're excited to see sort of high congruence, but I think it's important to keep that in mind that it is. I mean, I don't think AlphaFold2 is particularly overfit, but there is probably some overfitting, and I think it's important to do the standard practice of ensuring that when one is assessing these methods, is doing it in a way, is doing it on training data or sorry on validation data or test data that does not include in the training set, because we do see a delta. We do see a delta here, which is not insignificant. Okay, so let me sort of wrap up by just talking a little bit about so the implications and applications of this, so where I see this moving forward. So like I said earlier, without refinement, at least, actually, I didn't say this earlier, but without refinement, we don't have to do any conformational sampling. And so the speed up here is significant, right? It is on the order of 6 to 7 orders of magnitude. So this is not, this is a rather substantial speed up relative to anything. So for certain applications, right, so you're really going from days to milliseconds. For certain applications, like for example, docking, we think this could be very exciting because in those instances, right, you're often docking in a way in which the protein is held rigid, and then and you have a small molecule or something like that, which is allowed to be flexible, but the protein is held rigid, and so that obviously doesn't comport with our expectation that many particularly protein ligand induced binding does result in some conformational changes in the protein. But that's partly restricted because of the fact that predicting structures is very expensive. But if we're able to do this very rapidly, if we're able to essentially predict structures, conditional on a ligand, which we don't do yet, to be sure. But if we're able to do that while maintaining the speed, then potentially we could go much farther. But of course, the issue right now is that refinement is important for us. So one thing we're sort of working on to do right now is essentially trying to do refinement in a differentiable way to sort of get the best of both worlds, to get the speed as well as the performance. Another area which I think I'm certainly very excited about is modeling the effects of genetic variation. Right? So how do mutations impact the structure and then ultimately function? And this is an area where we haven't assessed our much as I should be upfront, but certainly no one's assessed AlphaFold2 either. So it's a bit of a gray area. We don't really know how all these methods work, but I suspect that having a method that is more sensitive to single sequences would be the way to go, because ultimately, at least in instances in which genetic variation leads to substantial changes, it's precisely those substantial changes that we're really interested in, right? Because it's those kind of not family average properties, or the aspects of the protein structure that are very much dependent on the specific sequence that's present, and the things that are not in the MSA in a way. And so this is an area where I think we could make some significant progress. And I mentioned this earlier. I mean, with viruses, we're in the midst of a pandemic right now. With viruses, certainly you have proteins that evolve very rapidly, and so even though they're not orphan proteins, but they may be moving so quickly, and the changes that occur during their evolution are precisely the things that we're interested in, not the parts that stay static, but the things that actually change. So this is another area where I think single sequence based methods will have a role to play. And of course, protein design, which I show you some results on. So I think I probably spoke a little bit too fast. I may be finishing early, but that gives us more time for questions. So just to all the people who did the actual work, Ratul, Nazim, Serjan, and Charlotte, who are both in my group, and so Ratul and Nazim are in my group and Peter Sorger's group. Charlotte is in my group, and Serge was in George Church's group before he moved on to found his own startup now. So really, the RGN2 work is all due to them. And yeah, I'll stop here and take any questions.
Thank you very much, Dr. AlQuraishi. That was really interesting. So if anyone has any questions, please feel free to write them up in the chat or in the Q&A, and I will be able to ask them for it. We don't have any yet. So now, forgive me for my ignorance. Protein modeling is something I was never really involved in, so this might be something that is known. But for training sessions, for rather training data sets, you had mentioned the one that you used was not the same as the AlphaFold. What makes for a good protein folding training session? What is the most important element that you're looking for?
I mean, I think so. That's an interesting question, actually, because when you're trying to build a general model for structure prediction, in some ways, what you want is to maximize diversity. You want things to be as different as possible, to sort of cover as much of structure space as possible, and that's what I think we'll all do. What I think, and I should say, I mean, the point I was mentioning where we didn't have it in the training set, the point there was really just to say that it's that has to do with the fact that we're not overfitting on it. So we intentionally excluded it so that we can assess the quality of the model on a set of data that we did not see in the model. I will say, though, moving forward, one area which I think would be interesting, just kind of thinking about, is constructing data sets that might be, say, more suitable for a given task, right? So maybe if you're very much interested in helical proteins, or in de novo design, or in a particular class of protein domains, then perhaps one can sort of construct data sets that are really focused on that, and allow one to essentially kind of train models that are more bespoke, that are not kind of general purpose structure predictors. And that's something that I think will be an area of interest moving forward. It's not something that I don't think anybody's done yet, but I suspect it will be an interesting area.
Neat. All right, so we do have some questions here. So our first question comes from Badri Adhikari. He asks, how will single sequence-based methods, for example, RGN, give an advantage towards explaining things such as genetic variation?
Right, so the idea here is a bit implicit, right? The idea is that with multiple sequence alignment, you're essentially taking a whole bunch of sequences and essentially averaging them, right? So if there are aspects of the structure that are really contingent on parts of the protein being different, those will be sort of averaged out, right? Because those are not the common part in an MSA. And so sort of MSA-based methods by construction will have a hard time kind of discerning those differences. While with single sequence methods, at least in principle, since you're always doing that full lift of going from the sequence to the structure, you never have that essentially that kind of crutch, that sort of, you can never lean on the multiple sequence element being present and sort of extracting that information directly from it. And so I think it's in that way that I suspect single sequence-based methods will be better for capturing genetic variation. But we haven't assessed it yet, and in part because it's hard to actually get data on this, where we have a lot of structures with slightly varying sequences. But if I think for to have that, we could more systematically assess these methods and potentially start building tools to get at this question.
Okay, and a follow-up that you touched on a bit there. Does RGN2 use multiple sequence alignment during its training?
No, it's not used at all. It's just simple sequences, yeah.
Excellent. Our next question, on the claim that protein folding is basically solved, can you please explain further?
Well, I mean, it's a bit of a contentious issue. So first, it makes sense to sort of step back and can define the problem, right? So protein folding, historically at least, really had to do with a much kind of broader category of problems, having to do with kind of, let's say, understanding the kind of the underlying physics of protein folding, in particular the dynamical trajectory that a protein takes to arrive at its lowest energy state, at its native state, right? So it's not just about the final structure; it's really about the trajectory. Like how does a protein go from a denatured protein to the final three-dimensional conformation? And simply that we don't have. We don't have a way to reconstruct that trajectory yet. What we do have is something that allows us to get the final structure, the native state itself. And there, I mean, so one can split hairs whether that problem has been solved, the protein structure prediction problem. I think for the majority of problems, it seems to be the case that it's yes, in the sense that when I say problems, I mean individual proteins on their own, not dependent on folding with other proteins, not in complex with other proteins or ligands and so on, and that do have a well-ordered structure. Those proteins do, as far as we know, it seems like AlphaFold2 is, and if you have deep MSAs, it does seem like AlphaFold2 is able to basically predict all of them, with pretty decent accuracy. But there are these wrinkles, there are these qualifiers, and I think, these qualifications are important. And I think, now that as we begin to think about protein complexes, particularly ones that, proteins that co-fold together, things of that sort, these are the new frontiers that we have to push forward that we haven't yet addressed with AlphaFold2.
Excellent. So our next question, it seems that CNN are out and transformers are in. Is there something similar to the idea of a BERT-like environment for proteins in 3D prediction?
So in terms of sequence space, that is essentially part of what we've done here, and other people have done as well, guys. So, RGN2 have AminoBERT. There's a bunch of other systems like UniWrap, ESM from Facebook, and ProtTrans, I believe, a few of those models, which basically essentially apply the ideas of self-supervised learning, like BERT, to the protein context on the level of sequence. I don't think we've seen those applied on the level of structure, but I think that's kind of a natural objection to go into as well, and that's something we're very much interested in. And so being able to essentially do self-supervised learning on structure, but I don't really haven't seen anything significant in this space yet.
All right. What is your opinion about using AlphaFold2 to model proteins with multiple chains using a polyglycerine linker to connect those chains?
Yeah, that's one of the tricks that people have been using AlphaFold2 kind of unofficially, and I think it makes perfect sense. I don't even think you actually need a glycine linker at this point, because one aspect of AlphaFold2 which is really interesting, right, is that it's it has a sort of positional encoding of where the residues are in the protein. But that encoding is actually fairly limited in that essentially it really only tells you how far apart any two residues are up to 32 positions, up to 32 residues apart. So beyond that, it's blind. It doesn't know how far two residues are. Yet it's able from that information to still recover structures. So it looks at structures in space and essentially says, all right, I know these two things ought to fit in some way, and even though I don't know how far apart they are, I am able to kind of recover their interface. This is for single proteins, but that kind of training regimen, the way it was trained, seems to allow it to now deal with protein complexes in a very similar way, because it's essentially, the way it's presented to it is that you're given two proteins that are sort of infinitely separated. But from this perspective, that's might just as well be 32 residues apart, right? It sort of can't really tell the difference. And because of that kind of ambiguity, people have been able to leverage it to actually predict interfaces of protein complexes. So I think it's cool, but I mean, I think it's important to keep some things in mind. I mean, one, obviously, first it wasn't trained for this purpose, so I think the performance is going to be somewhat unpredictable and probably, subpar relative to what it can achieve if it were trained for this purpose. And two, while there are some similarities between protein-protein complexes and sort of intra-proteins, say surface binding or surface interactions, they're not entirely identical, right? I mean, in the fact that within a protein everything is covalently linked, it still plays some role in terms of avidity effects, at least. So biophysically, there's probably some delta between the two things. They're not quite the same thing. And so I do think, AlphaFold3 or whatever, in a couple of years from now, I think we'll have much better tools to do this in a much more sort of directed way. I think what we have right now are basically kind of hacks that do this because it's sort of available and it's possible to do it. But I don't necessarily think it's the best way to do it.
That makes a lot of sense, actually. Can you offer any insight into the biological importance of a specific residue or a set of residues by modifying input sequences, comparing predictions, or some other kind of sensitivity analysis?
I think so. We haven't done it yet, but that is the sort of thing which I think we ought to be able to do. It's a sort of analysis which very much lends itself to this kind of architecture. It's even conceivable to think about ways in which one can actually try to understand through the network what is it about that change that alter the structure, right? So you can imagine, hypothetically, let's say if some residue changes and then the structure changes in a major way, presumably there will be sort of signatures in the neural network that sort of identify how the neural network kind of came to make this move. What other residues did it look at? Ones that may be involved in the change, but maybe ones that are not involved in the change. That drove this shift. And so that's definitely the kind of thing which I think we can do. And more broadly, I would say, I mean, even beyond our model, I think there's a lot to do in terms of probing AlphaFold2, for example, or RosettaFold, other approaches, and trying to understand how they work and why they're making the decisions that they're making. This to me, I think, is sort of a fairly, I think, actually broad space to operate on, and I don't see much activity there, but that I think is a really interesting direction to go into.
All right. Our next person opens by thanking you for the fantastic talk. And then they ask, do you think providing sequence variations present in the human population with some annotations, like whether the variants are known to be pathogenic or not, do you think that would help in assessing the impact of genetic variation?
Possibly. I mean, it's sort of interesting. I mean, I think yes, but I think we have to show this, right? It's sort of because it's there's been this dogma in the field for a long time, right? That sequence determines structure and structure determines function, and so if you have structure, we ought to be able to infer function. But we've never really been presented with this kind of task in a serious way because we didn't have structures. Structures weren't very abundant, and in fact, in instances where we did have structures, those proteins were typically well characterized enough that people had also functional assays on them. Now we're in sort of the situation where we're being asked, can we essentially infer function from structure in a systematic way? And that, I think, remains to be seen. And I think will require some degree of tool building to make that association. Now, I'm optimistic. I think it can be done, but I don't think it's yet been done.
Okay. Just a follow-up on that. What would be the biggest hurdle that we still have to overcome for that to become a reality? Because you said you're being optimistic about it. What would make that a much more realistic prospect instead of an optimistic one?
So I think the rate limiting factor here, so there's one issue, right? So unlike structure or sequence, function is very kind of multimodal, right? The notion of function is a bit ill-defined, and in some ways depends on what it is you mean, right? Or what it is that you're trying to measure. You have enzymes, you have proteins that are structural, you have proteins that are much more about motions, you have, right? So there's quite a bit of variability into what proteins do, and there are a lot of assays that people use to assess protein function, unlike the situation in sequence and structure. So I think the challenge here is going to be enough data that and sort of really kind of meaningful definitions of function that allow us to get at this question. For clearly some aspects of protein function, like people have done these deep mutational scans, there we have data and the function is well defined and very circumscribed, and there I think it'll be fairly straightforward. I mean, there's already work in the space. But I think having a kind of let's say a broader, a more kind of general notion of protein function and being able to kind of really take any given protein and say, okay, what does this do in its context in the cell? That I think will require a fair bit of work because it just depends on so many other things and depends on having much more data sets that would have much broader coverage of function, which we don't yet have.
Okay. So one last question here. Say RGN2 achieves a higher than AlphaFold2 accuracy. What's next?
Next, I think there's multiple next. One is different conformations, so being able to predict conformational changes, and not just the ensemble of conformations that a protein has. Protein-protein interactions, protein complexes is another obvious thing, and that thing is very important. And a bit longer term is beginning to think about how these things assemble into molecular machines, right? So not really just about kind of ensembles of different conformations, but really thinking about kind of correlated motions and how these things may actually operate in a cell. If you think about like myosin or something like that, a complicated beast that has multiple proteins in it and that are all moving in a specific directed way, that remains way out of the realm of the possibility at the moment. But that I think it remains kind of the key block before we're then able to, I think, put these things together and start thinking about actually doing something like a cell simulator. But that's the ways up. But those to me seem like the obvious next steps.
I can see that would be an amazingly complex but amazingly valuable tool. So I miscounted. There is one more question. Sorry. How accurate is RNG2 or AlphaFold in terms of predicting disordered, that is, unstructured protein regions?
Yeah. So I can't really answer that for RGN2 because we haven't assessed it systematically. For AlphaFold2, there's a bit more evidence for this. It seems to be a pretty good disorder predictor, but probably state of the art, better than anything else, or at least as good as a state of the art. The only issue is the way this works. So AlphaFold2 does not have an inherent disorder predictor, but what it does have is a predictor of essentially a self-assessment of how reliable is a given residue prediction. And based on that, essentially people have taken that, including DeepMind, to sort of, be correlated with disorder. That if AlphaFold doesn't know what this thing looks like, it probably is disordered. And that is true, but it is confounded because it may also simply because AlphaFold doesn't know what this is, not because it's disordered, right? So there's this kind of funny conflation. So I think, again, this is one area where I think I expect we'll be able to make a lot of progress just by disentangling those two sources of uncertainty in AlphaFold2. But it's sort of, it's not, it's not solved yet. And I should say, beyond, so, people so far think of disorder as a sort of binary thing, either it's ordered or disordered. But I think what's going to be more interesting, moving forward is really thinking about ways of parameterizing disorder, right? It isn't either or. It's probably some sort of continuum, and then there are probably ways in which two different disordered regions are kind of meaningfully different, and there ought to be ways for us to kind of characterize that difference. And I think this is new conceptual ground, so I think it's exciting because we'll have to kind of define the question, not just try to answer it. And that to me, I think will be an area of growth as well, in the future.